Thank you for your attention. I'd like to thank the organizing committee for inviting me to share some of our work that we've done regarding bacterial keratitis. And this type of infection primarily affects extended use contact lens wearers, as well as those who have undergone refractive corneal surgery and patients who are immunocompromised secondary to conditions such as diabetes, alcoholism, malnutrition, and steroid use. In 2005, it was reported that there were 18 cases of microbial keratitis for every 10,000 contact lens wearers in the United States. And of these patients, about 20% of them had some level of visual acuity loss. And so there's the loss of vision, as well as substantial economic impact from this disease, which you can see by the $15 to $30 million per year in treatment costs. Clinically, this type of infection presents as a rapidly progressing suppurative stromal infiltrate with a marked mucopurulent exudate. There are common features of this type of infection, including yellowish coagulative necrosis of the cornea, ulceration of the stroma, perforation of Descemet's membrane, and then ultimately one can get perforation of the cornea as well. And because of these structural changes to the cornea, you can also get secondary development of cataract and glaucoma as well. So the leukocytic infiltrate into the infected cornea includes neutrophils or PMNs, and these cells will extravasate from the vasculature and migrate to the extracellular matrix, where they carry out their primary role, which is to phagocytose invading pathogens. They also release antimicrobial peptides such as defensins from their cytoplasmic granules during the process of degranulation, and then they utilize extracellular traps or nets to immobilize and kill invading pathogens. They also release cytokines that help to modulate the inflammatory response as well. In addition to PMNs, another essential cell type is macrophages. These antigen-presenting cells assist in debridement of apoptotic and necrotic tissue, also removal of pathogens. They, too, release cytokines as well as growth factors. And Dr. Linda Hazlett's group has shown that macrophages regulate the influx of PMN into the infected cornea as well. And then, despite the fact that the cellular response is predominantly neutrophilic, you do see CD4 positive T cells later in the immune response as well. In addition to the cellular response, we are also interested in corneal nerves. Here we have a couple images illustrating a normal human corneal neuroanatomy. And here we can see structure A in the upper left-hand figure, and then the blue fibers in the lower image. These nerves are comprising the subbasal plexus that overlie Bowman's membrane, and then you can see that they're going to send up these perpendicular beaded projections that traverse through the epithelial layers, reaching the corneal surface. These branches can be either sensory, sympathetic, or parasympathetic, and they have a variety of functions, including corneal sensory perception, as well as delivery of neuropeptides such as substance P and VIP. And our group is particularly interested in VIP, or vasoactive intestinal peptide. This is a 28 amino acid neuropeptide. It has a short half-life of about 2 minutes. It's delivered by a number of different, neurons, as well as being produced by a number of different organs, such as the gut, the pancreas, the suprachiasmatic nuclei of the hypothalamus. It's also been shown to be produced by immune cells, and its functions range from relaxation of smooth muscle cells to lowering arterial blood pressure, also vasodilation, as its name implies, and it has a number of immunological activities that are carried out by, in particular, 2 receptors, VIPR1 and VIPR2. And so we study bacterial keratitis using mouse models of disease, where we can infect the cornea using Pseudomonas aeruginosa. We chose Pseudomonas because it is one of the most commonly isolated pathogens in bacterial keratitis. In fact, it makes up about 70% of cases related to extended wear contact lens use. And it's a gram-negative opportunistic rod, and basically, depending on the type of mouse that we use, we can get one of two responses. So if we infect C57 Black 6 mice, which are typically categorized as Th1 responders, the cornea perforates, and this would be our susceptible phenotype. If we infect the BALB-C mouse, which is defined as a Th2 responder, the cornea tends to heal by 7 days post-infection, and this would be our resistant phenotype. So we first looked at VIP expression in the corneas of both BALB-C and B6 mice. And what we found was that the distribution patterns of VIP-ergic fibers under normal conditions were pretty similar between both strains. And then in regards to B6 animals, this staining pattern didn't really change over time after infection. However, in the BALB-C corneas, we found that staining became more intense along the corneal epithelium and throughout the corneal stroma over time after an infection. And then this was further corroborated by protein expression as detected by EIA. So we next sought to perhaps convert the otherwise susceptible response of a B6 mouse into one that resembled something more resistant by injecting these animals with VIP. And what we found, first characterizing the response by clinical score, is that at 3, 5, and 7 days post-infection, the VIP-treated animals had less severe disease when compared to the PBS-treated animals. And this is illustrated by the slit lamp photographs shown here at 7 days post-infection. We can see a typical response in our PBS-treated animals. The cornea is perforated. This would be what we call a +4. And then after VIP treatment, we can see this would be considered a +2. We next looked to examine the effect that VIP had on a number of different pro and anti-inflammatory mediators that we know are important in this type of infection in our mouse models. So at 7 days post-infection, we took a look at a number of different pro-inflammatory molecules, interferon gamma, IL-1 beta, TNF alpha, and MIP2, and they were all significantly downregulated with VIP treatment. In contrast, the anti-inflammatory molecules that we typically look at, TGF beta 1 and IL-10, were significantly enhanced after treatment. And then again, this was confirmed by looking at protein levels for IL-1 beta, MIP2, and TGF beta 1. At this point, we then looked to see if there were any changes in bacterial load in these corneas. And in fact, at 5 days post-infection, we did see that VIP treatment decreased bacterial load in the corneas. And then we also saw a reduction in nitrite levels, indicating that macrophage activation was decreased, and a decrease in myeloperoxidase levels as well, which corroborated our histopathology sections showing that there was in fact a decrease in PMN infiltration into these corneas. So VIP is known to be a macrophage deactivating factor, so we next isolated macrophages from both BALB-C and B6 animals. We stimulated them with LPS, which is a very potent virulence factor that is part of the cell wall of Pseudomonas, and looked at what happened in the presence of different concentrations of VIP and measured expression levels of IL-1 beta and MIP2. And the take-home point here is that LPS stimulation, as expected, upregulated production of both IL-1 beta and MIP2 in both BALB-C and B6-derived macrophages. But if you see here, VIP was able to dampen that effect in BALB-C animals, but if you look at B6, it was also able to dampen the effect, but only at the highest concentration that we tested. And this similar trend was observed when we looked at PMNs and MIP2 expression. So we next asked, does this disparate expression in IL-1 beta and MIP2 in response to VIP have anything to do with the VIP receptor being expressed on these cells? And so again, we looked at macrophages and PMNs from both B6 and BALB/C animals. And what we found was that under normal conditions and after LPS stimulation, we were able to detect VIP-R1. But if you look, BALB/C animals express much higher levels of this receptor when compared to B6, at least as far as the macrophage is concerned. When we looked at PMNs, there was no difference in VIP-R1 receptor expression. So we then asked, is there any role that VIP perhaps is having, like is exposure to VIP perhaps enhancing VIP-R1 expression on the macrophage? And so we used confocal laser scanning microscopy, looking at B6 corneas at 5 days post-infection. And what we found was that PBS-treated corneas stained positive for F480, which is our macrophage marker, but very few of these cells co-expressed VIP-R1, one of which is shown in the inset here. On the other hand, though, after VIP treatment, we saw that we had a number of macrophages that stained positive for both F480 and VIP-R1, indicating that just the presence of VIP was able to enhance the receptor being upregulated, and perhaps we think that this is because it's another way for VIP to augment its functions. So in summary, some of the factors that influence the development of the resistant versus susceptible response in regards to VIP includes the availability of endogenous VIP, where BALB/C mice have a higher level of nerve fiber distribution as well as protein expression. And then the receptor expression on immune cells themselves, where, again, BALB/C macrophages have a higher expression of VIP-R1, and they can more efficiently upregulate this receptor when stimulated with LPS. And then the overall sensitivity to VIP, where B6 animals are less sensitive and therefore require higher endogenous levels of VIP to get the same response. We then took a look at VIP's effect on adhesion molecules and extracellular matrix, and I'm just going to point out one of the particular studies that we did, or one set of results that looked at ICAM1 and its counterreceptor LFA1. And we chose ICAM1 because previous studies from our lab have shown that when you knock out ICAM1, there was a very significant role in this molecule in recruiting PMN into the cornea. This is just a PCR array giving you an idea that, in fact, VIP does have an effect on a number of different adhesion molecules and components of the extracellular matrix. But the ICAM1 LFA1 interaction that I was talking about, we performed immunofluorescence staining for both of these molecules. And if you look at the PBS corneas here, what we see is positive staining for ICAM1 in red, LFA1 is in blue, and so their interaction shows up in pink. And that interaction basically is indicative of active transmigration of inflammatory cells throughout the cornea. So we see there's a lot of migration going on here. And then you can appreciate the very dramatic decrease in ICAM1 expression as well as LFA1 expression. And instead, we are left with a much more intact epithelium after VIP treatment. This is just summarizing some of the other results that we found in regards to adhesion molecules and extracellular matrix components. Basically, we found a downregulation of the components that were known to kind of be pro-inflammatory in nature and break down the matrix. We also saw a downregulation in other adhesion molecules, while those molecules that were associated with corneal restoration were upregulated. And then the take-home message with this slide is just we're localizing VIP-R1 in BALB/C and B6-derived fibroblasts as well as VIP-R2. And what we found was that VIP-R1 was not localized, but rather detected throughout the cell body of fibroblasts under normal conditions, when they were exposed to VIP only, LPS only, or a combination of VIP and LPS. So basically, their expression was throughout the cell body regardless of conditions. On the other hand, VIP-R2 was localized to the nucleus under normal conditions and really only was translocated into the cell body upon VIP exposure. And this set the framework for us to continue studies and find that, in fact, VIPR1 seems to be the receptor that carries out VIP's anti-inflammatory functions, while VIPR2 seems to carry out its healing functions and functions to reconstitute the cornea. So to summarize, it's the unregulated and persistent inflammatory influx, particularly by PMNs, and then the subsequent proteolysis of the matrix are major causes of bystander tissue damage during Pseudomonas-induced ocular infection. And that VIP influences not only immune cells, but also the microenvironment, as we saw with downregulation of many of the adhesion molecules and extracellular matrix components. And that between VIPR1 and VIPR2 receptor expression, that VIP is able to function as a true immunomodulator, permitting inflammation to occur with expedient transition to the restoration of immune homeostasis. We've also had a few additional studies that have shown that VIP excuse me, not only affects growth factors, but also angiogenic molecules and defensins. We've also shown that it is able to upregulate anti-inflammatory toll-like related molecules such as SIGR, while down-regulating TLR4, which is the major toll-like receptor for LPS. And then essentially this slide kind of summarizes all the effects of VIP thus far. So we see a downregulation of the pro-inflammatory factors, upregulation of anti-inflammatory factors, which ultimately deactivate macrophages and prevent the persistence of PMN in the infected cornea. And so with that, I'd just like to acknowledge the people in the lab, Tommy Carrion, Cody McWhirter, who carried out a lot of this work, as well as the lab of my former mentor, Dr. Linda Hazlett, and then Ron Barrett and Carrie Vistison, who are members of the core grant that Wayne State has. And I'd be happy to answer any questions.
Any questions, please?
Any questions? Right.
How do you think the bacterial load was reduced?
That's a good question. So we do get some activation of macrophages. It's just not to the same extent that we would get otherwise in the B6 cornea. So there is some pro-inflammatory response that does occur. It's just not as dysregulated as it would otherwise be in a normal B6 response. So the nice thing about VIP is that we find that 3 days is really the tipping point for its effects to kick in. So up until then, the response that we see at like 1 day post-infection, for example, is exactly the same as if we weren't treating it with VIP. But once 3 day comes, that's where we see it really diverge and take the healing route versus the dysregulated pro-inflammatory route.
So in histology of the cornea, we can find a lot of macrophages.
We do find quite a few macrophages, not nearly as many as neutrophils, though.
So immune system is working there very actively, or?
Oh, yes.
Oh, okay.
Are you talking about after VIP treatment?
No, in cornea.
In the cornea in general?
So if you have to treat VIP to the cornea, how will you treat? As a eye drop, or you are injecting? What are you going to do?
So in this particular model, these are IP injections, and it's only of a 5 nanomolar concentration of VIP, which is very, very small, especially when you consider the half-life. So it's definitely setting off a whole stream of events. But now we're working on topical application so that it's a little more clinically applicable, because obviously no one wants to come in for injections.
Thank you.
Mm-hmm.
Thanks. Very nice.